And we will also, like me and other tutors, TAs that will be taking these sessions, will upload these on you will receive links via NPTEL also, and we will upload these on our own YouTube accounts as well. Okay. Hello, everybody. I will give you a brief introduction about myself. Okay, let me share the screen first. So as I share my screen, I can converse with you guys as well. I hope most of you might have attended other sessions on NPTEL. If not, if this is your first session, I hope it will be a good one. It will be a good start for you. Just a moment. Yeah. So I am sharing my screen. I hope it is visible to all. I will stop sharing the screen in between if any of you guys are trying to join, right? So in that case, I'll stop the screen sharing. I will admit the people who are trying to join, and then I'll share it again so that there is a minimum like fault on this technical end. Uh, yeah, some people I can admit while sharing as well. Okay. So here is a brief introduction about myself. I am Arveen Kaur. I am a third year PhD scholar in the Department of Biological Sciences and Bioengineering at IIT Kanpur. And presently, I am the TA for this course, that is Cell Biology, Cellular Organization, Division and Processes, that is being conducted by our professor, Professor Shikha Laloria, who is a professor at IISc Bangalore, another very renowned institute. So you might have already gone through her first week's lectures. Uh, Let me briefly introduce as to how I will proceed with these sessions that we are going to have for the next 2 months, right? So this is the first session out of 8 sessions that I am going to conduct. I will briefly introduce as to what I'll be doing in these sessions. So how I will proceed with it is, for the first part of the session, I will discuss the previous year's assignment for that particular week's content. Right. So for example, in this week's content, there was a brief introduction about cellular cell biology, and then there was a brief introduction towards the genetic material, that is DNA, that was given to you guys by Professor Shikha's lectures. So, and you have also received an assignment that I think is due by the first week of February, if I'm not wrong. Right. So, since the due date of that assignment has not passed yet, I will not be discussing that particular assignment today. Right. Once the due date for that assignment is over, if you have any doubts with the answers, once they are out, or with any question, if you are unable to understand it, I will be more than happy to assist you with that concept. But for now, since it is an assessment, right, of what you have understood out of the lectures, I will not discuss that same assessment until its due date is over. But on the other hand, what we can do is that NPTEL has provided us with the previous year's assignments for the same weeks, right? For all 8 weeks, I have the assignments from last year's course as well. And there might be a few overlapping questions between that year and this year. I think there is no overlapping question in week one, as far as I have gone through this assignment. So what I am going to do every week, yeah, yeah. Is anybody trying to say something? Okay. I hope my screen is visible. Is it?
Yes.
Okay. Yes, ma'am. Okay, cool. So, like I just told you guys, what I'll do in the first half of these lectures is that I will discuss this previous year's assignment, and it might contain some concepts, if not the questions themselves, that might overlap with concepts that will help you to solve this year's assignment. Also, along with that, I will dedicate like half the lecture to some doubts that arise. Uh, just a moment, I will have to go back to the lobby to admit people. Okay. Yeah. So half the lecture I'm going to dedicate to some additional concepts that I might have felt were looked over, because it is a concise course. All NPTEL courses are very concise courses, right? So sometimes what happens is, because there is such a vast syllabus to teach, some basic things the professors might think that you guys already are aware of these things, right? But uh, since we have this additional time, we will try to deal with those things that have been just like looked over at that in those lectures, right? Just because of the unavailability of this excess time at that place, right? So that is another thing that we are going to do. And in that particular session, what I will also do is, I always, before like coming to this lecture, or every Sunday, I go through the discussion forum, and whatever doubts you guys have posted on the discussion forum, I try to basically go through those doubts, and I try to incorporate the concepts that could clear those doubts in that particular half of this session, right? So that is another thing that we are going to do. So let us begin with the first week's previous year's assignment. Uh, okay. So what I can do in these 10 questions is, how I have prepared it is, I will first read out the question. I will give you a little time if required to ponder upon what the answer might be, and then I will discuss both the answer as well as the concept behind the answer if it is not like clear already, right? Uh, yeah, I've admitted everybody. Okay, so the first question being, gram-positive bacteria are stained purple with crystal violet because they have thick peptidoglycan layer and no outer membrane. B, they have thin peptidoglycan layer and no outer membrane. C, they have a thick peptidoglycan layer and a thin outer membrane. Or D, they completely lack the peptidoglycan layer but have an outer membrane. So think about the answer. It was one of the concepts that was primarily dealt with in Professor Laloria's lecture. This is just to brush up your lectures, right? Meanwhile, let me just admit some people. Okay. Yeah. Okay. So let us move on to the answer. I expect a lot of you to already know this answer, which is A. That is, uh, okay, there are some people who are trying to join. Which is A, that is, they have a thick peptidoglycan layer and they have no outer membrane. So this is the gram-positive bacteria. So once we use crystal violet staining, this purple colored stain is what we see in case of gram-positive bacteria, and this pink colored stain is what we see in case of gram-negative bacteria. So why is it so? Because of this reason that we talked about, that this peptidoglycan layer is thick and it is present in gram-positive bacteria. It is also present in case of the gram-negative bacteria, but it is very thin. And in addition to this peptidoglycan layer, they, ha they have an outer membrane, right? This outer membrane has certain lipids, carbohydrates, etc., due to which the same stain, that is crystal violet, gives the pinkish hue in this case, right? So, yeah.
Mm. I'm
Yes,
sorry to interrupt, but the screen is not visible.
oh, it's not visible. Okay. Okay, that happens once. Um, okay. I'll share it again. So once I'm trying to admit people in between the lecture, this is a problem that I have faced before. So I will keep this in mind. I will try to stop sharing and share again. Okay, just in case you guys couldn't see the question earlier, let me go through it very quickly again, which is that whether gram-positive bacteria are stained purple with crystal violet. Why is it so? So our answer to this was because they have a thick peptidoglycan layer and they lack an outer membrane, right? So this is the image that I was showing, a representation which showed that the gram-positive bacteria, when we stain it with crystal violet, it gives a violet colored staining only. But because of the presence of this outer membrane in the gram-negative bacteria, which is absent in case of gram-positive, it gives a pinkish stain in case of gram-negative bacteria, the same stain, crystal violet, right? So, also over there, I explained that this outer membrane, why is it giving a different property to the stain altogether? Because it has different components than that of the peptidoglycan layer. So, peptidoglycan layer, as its name suggests, is consistent of peptidoglycans, which are protein and sugar molecules, right? While in the outer membrane, there are different kinds of macromolecules that are more abundant, which are lipopolysaccharides and other carbohydrates, right? So, which is why the property of the staining is also different in case of gram-negative bacteria. Moving on to question number 2, which is which of the following statement doesn't hold true for the nucleolus? So nucleolus, as you all might remember, is one of the components of the nucleus. Uh, okay, just hold on a second. Let me admit these people. Okay, I've stopped sharing. Let me share again. Yeah, okay. So which of these is not true for nucleolus? Okay. It is a dense body present inside the nucleus. It is a site of rRNA synthesis. It is bound by a thin membrane, or it is a site for ribosome assembly. So this is another thing that was discussed very briefly, but all these points were discussed in the lecture that you all might have gone through. And let's not wait for a longer time. Let's just skip to the answer. Yeah, which is that it is bound by a thin membrane. All the other things that are written about the nucleolus are true, that it is a dense body that is present inside the nucleus. You remember ma'am telling us that it is a perinuclear dense body that is present inside the nucleus. It is a site of rRNA synthesis, and ribosome assembly occurs inside the nucleolus. But the only thing that was not discussed in the lecture is about it being bound by a thin membrane. So this is the answer over here. So over here, I have kept a representation of the nucleus, which also state all these um, points that we have just discussed, that it has a granular component wherein there is pre-ribosome assembly that occurs. Along with this, it has a fibrillar center wherein the synthesis of rRNA or transcription of rRNA is taking place. And it also has a dense fibrillar component. So two types of fibrillar components are there, a center and a peripheral dense fibrillar component wherein processing of this pre-rRNA takes place, right? So question number 3. Okay, since it is the first class, uh, it is okay that a lot of people are coming in late and trying to join constantly, but it will constantly create some kind of interruptions in the class, right? So I have admitted these people again. Let's come back to question number 3, which is the cell membrane is termed as a selectively permeable membrane because it allows the free diffusion of gases, hydrophobic, and small uncharged polar molecules such as water, and it also contains transporters to selectively transport large polar uncharged molecules such as glucose. The second one being the only way to cross the cell membrane is via the transporter proteins, or it is completely restricts the movement of large polar uncharged molecules across itself, or none of the above is true uh, for this selective permeable property of cell membrane. So which one do you think is the right answer? I think it is pretty simple over here. This is a concept that we have been studying since 10 plus 2, right? So the first one is the correct answer, that although it allows free diffusion of small molecules, hydrophobic molecules and gases, uh, but it does contain some transporters for larger molecules and for hydrophilic molecules, etc. Right? Polar molecules, charged molecules, ions, etc. Again, we have to pick the correct statement out of this. So, guanine is a purine base, thymine is a purine base, cytosine is a purine base, or adenine is a pyrimidine base. So, which one of these do you think will be the correct answer? This is also a concept that we have been studying for a while now. If you had biology in 10 plus 2 as well. So it will have been a time since you are studying this concept, but nonetheless, if not revised, people tend to forget it. So the answer over here is that guanine is a purine base, right? So purines have this 6 plus 5 membered rings while pyrimidines have 6 membered rings. There are two types of purines that are present. One is adenine and the other is guanine, right? While there are three of pyrimidines that we are aware of. One is cytosine, the other is thiamine, and the third one is uracil, which is present instead of thiamine in case of RNA, right? So these are the pyrimidines and these are the purines. So just for your knowledge sake, I have included this particular image. You can read as to what kind of a compound the purines are and what kind of a compound pyrimidines are. Okay, moving on to question number 5 for now. Okay, yeah. So question number 5 talks about another very talked about rule, which is the Shargaff's rule, right? Shargaff's rule states that the ratio of amounts of A to G and T to C are close to unity in DNA, or the ratio of amounts of A to C and G to C are close to unity in DNA, or the ratio of amounts of A to T and G to C are close to unity in DNA, or the fourth option, that is ratio of amounts of A to A and T to C are close to unity in DNA. So which one of these do you think is the correct option?
Ma'am, option C.
Option C. Okay. For this, I am going to give a little more time. Uh, I will just stop sharing and share again because I had come to admit people. Yeah. Yes. Okay. So one of the answers is option C. That is a ratio of amounts of A to T and G to C are close to unity. So let us see what the correct answer is. Oh, I have not marked it over here. Yeah. So the correct answer is in fact that A to T and G to C are close to unity in DNA, right? This one is the correct option. I forgot to highlight it over here, but I have made this representation on the side. I hope it will be clear for you guys. I will still mark it. that option number C is correct. Along with that, so this is rule number one of Chargas, right? So why is it so that A to G and A to T and G to C should be equal? Why not any other? You can think about it. I am anyway going to answer it, right?
Ma'am, because A
Ma'am,
bonds with T and G bonds with C.
Yeah. So uh, there is another rule that A is going to form a double hydrophobic bond with T, while G is going to bond with C. Okay, since I'm using my mouse, please excuse this handwriting. And G is going to bond with C via a triple bond, right? So for this purpose alone, it is compulsory that a number of adenines are equal to number of thymines and number of guanines are equal to number of cytosines. Along with that, there is another rule, which is like Sharka's rule, has two statements. One is this that we have just talked about. The other statement is that it is not important that the number of A to T is equals to the number of G to C. Right? Is that true? Yeah.
yes,
Because
ma'am.
this number uh, not only varies from organism to organism, it can even vary within the organism in different types of cells, right? So the number of the ratio of that A and T that is present is to G and C that is present is actually going to determine the type of folding that any kind of protein or any kind of biomolecule is going to have, right? Because this will more GC ratio will have a more compact nature, while more AT ratio might have a more expanded nature, right? So this ratio is never going to be same. It is going to differ not just within uh, different organisms, but between different organisms, but also within a single organism. Right? Okay. Moving on to question number 6. Yeah. So, which of the following organisms belongs to the prokaryotic? So, what are prokaryotics? This is what we are going to discuss after the answer. So, is it yeast or animal cells or archaea or protists? Which one
Ma'am,
of these?
Art.
Which
option
one?
C, Archaea.
Yeah, yeah. So that is actually the correct option, archaea. Right? So prokaryotic cells, what is a prokaryotic cell? A prokaryotic cell is a cell that does not have a defined nucleus. To begin with, the first statement, which or the first difference between a eukaryotic and a prokaryotic cell is going to be the presence of an undefined nucleus or a DNA or any genetic material which is not contained within a defined nucleus, right? So that organism is going to be prokaryotic. Instead of a nucleus with a well-defined cell wall, well-defined membrane, it has a nucleoid, right? Also, just like the eukaryotic organisms, it also has cytoplasm and a plasma membrane. And like some eukaryotes, it also has a cell wall. Apart from this, there are some cell organelles that are present, like ribosomes, but most of the cell organelles that we see in a eukaryotic cell, such as the Golgi apparatus or the cytoskeleton or the endoplasmic reticulum, lysosome, plastids, such cell organelles are absent in case of a prokaryotic cell, right? Also, another feature in many prokaryotic cells is the presence of flagellum or a pili or some cilia, which basically, because these are mostly unicellular organisms, so these structures help them in mobility, right? So this is also often present. Yeah. Okay. Okay, moving on to question number 7, which is which one of the following is true regarding carbon atom? So, does carbon form unstable carbon-carbon bonds? Or carbon can form 4 covalent bonds? Or there is an obvious upper limit to the size of the rings or the chains formed by the carbon atoms? Or all of the above? So, again, one of the major uh, molecules we talk about in biochemistry is carbon, right? It makes up all the biomolecules. It is a constituent of all the biomolecules, uh, most of them. And the answer over here is that carbon can form 4 covalent bonds. How you can narrow your answer down to this one correct statement is because of the first statement that is present over here, which is carbon forms unstable carbon-carbon bond. So this is a very basic knowledge that the carbon-carbon bond that is formed is a very stable bond. Carbon-carbon can form a single, double, or a triple bond, and these bonds are very, very stable, right? Yeah. So thus the answer being carbon can form 4 covalent bonds. Question number 8 being, which of the following allows the ions to diffuse down the concentration gradient? Is it ion transporters or ion channels or lipid bilayer or all of the above? So over here, we are just talking about the transport of the ions down their concentration gradient. So which one is the most likely answer? So the answer over here is ion channels, right? Ion channels are a very common constituent and one of the major constituents of the plasma membrane, right? These ion channels, as their name suggests, will allow some specific ions to pass through the membrane. So how, what do these ion channels look like? If this is a plasma membrane, right, then there will be a channel over here, right, which will be interrupting this plasma membrane. And then the plasma membrane can continue. And there might be another kind of plasma, this ion channel that might be present. And then the plasma membrane can again continue, right? The density of the ion channels is also variable in variable cell types, and which kinds of ions can pass through them is also different, right? For example, there is a specific kind of ion channel which will allow calcium to pass through it. There is another specific kind of ion channel which will allow potassium to pass through it, and yet another one which will allow sodium to pass through it. Now, all of these are positively charged ions, right? All of these are positively charged ions. They are either monovalent or divalent. So why is it so that uh, maybe a channel that is specific for sodium cannot let potassium pass through it, or a ion that is maybe specific for chloride cannot let calcium pass through it, right? So why is it so? Because these ion channels also differ in properties and they are very selective in the molecule that they are going to let pass through them. And how are these properties that we talk about different over here? Just a minute. Let me admit the people who are waiting in the lobby. Okay. Yeah. So I hope it is still visible. It is. Okay. So they are going to differ in their property, right? For example, I have kept the example of some ion channels which are voltage gated, some that are ligand gated, and some other which are tension gated or mechanical ion channels. Sorry. So what are these ion? How are these ion channels different? They are different because the voltage gated ion channels, what are they going to do? They are going to sense the difference in the voltage between the inner membrane and the outer membrane region, right? This is the plasma membrane. Now, inside the membrane and outside the membrane, they are going to sense a temperature difference. For example, say that the inside is more negative and the outside is more positive. Now, they want to, yeah. Now they want to the cell to get depolarized, right? How is the cell going to get depolarized? When it is going to gain positive charge, right? So for this depolarization to occur, now this channel might open up and it might allow influx of ions such as calcium, if it is a calcium ion channel, potassium, if it is a potassium voltage gated ion channel, or sodium, if it is a sodium voltage gated ion channel, right? So whichever ion it might be specific for, it will allow the influx of that particular positive ion such that the depolarization of this membrane can occur. And now after the depolarization has occurred, there will be more positive charge inside than there was before, right? And this was the purpose of this ion channel to open up. Similarly, there are other kinds of ways through which these ion channels might work. One of them might be ligand gated channels. For example, we say that some molecule has to pass through this ion channel, but this ion channel has a morphology where it is closed, right? It is physically unable for any ion to pass through it. But this ion channel might have specificity for binding of some of the ions, right? When these some ions come and bind to some particular sites of these ion channels, it might open up this channel, right? And thus it is known as ligand gated channel. And then there are also mechanical gated or tension gated channels that are present. How these work is that there are some tension or mechanical cells that are present in its vicinity. Now, when these mechanical cells change their structure, they pull the channel apart such that whatever ion has to pass through this particular channel can go and go into the membrane, right? One example of a mechanical gated channel is, I think, the potassium channel present in some bacterial populations, right? So, yeah. So this is how ions are allowed to diffuse down the concentration gradient. Also, another question that I have linked to this one is that once this is a statement that is said, what kind of a transport will this be? If you guys understand my question, if somebody says the statement that ions are diffusing down the concentration gradient, what is the kind of transport that is occurring? Is it an active transport? Is it a passive transport? Is it a diffusion? What is it?
Ma'am, active transport.
So no. Okay. Active transport is generally against when there is movement of ions against the concentration gradient. Usually then ATP has to be used, and that generally occurs in case of sodium potassium pumps or any other pumps, right? Wherein there is movement of ions or other molecules against their concentration gradient, right? So for that, there has to be additional force that has to be applied. But when it is down their concentration gradient, that is, that calcium is abundant outside the cell and it is moving inside the cell where it is very little, right? With respect to its availability outside the cell. So although it is using this particular channel, it is following passive diffusion. There is no ATP that is being used up in this particular process, right? There are other mechanisms that are happening. That is, the gate is being opened up. That is because of either the voltage difference or the structural changes due to ligand binding or tension, cells contracting, right? Just because these processes are happening, it is not important that ATP is being used up. So that is another point, right? So this is also a passive transport. Okay. So moving on to question number 9. The bond between nitrogenous base and the sugar group in the nucleotide is termed as which one of these? Phosphodiester linkage, N-glycosidic bond, O-glycosidic bond, or hydrogen bond. So this is one of very important questions, right? If you have read this concept of the bond formation in DNA molecule thoroughly, you will know that there are multiple kinds of bonds that exist between different DNA molecules and sugar groups and nucleotides in the phosphate backbone that is present, right? So which particular bond is being talked about over here in case of one between a sugar group in the nucleotide and the nitrogenous base? So think about it.
Bond.
Yeah, which one?
Phosphodiester bond.
Okay. That is the one that we have most heard about, right?
Yes.
So is it a bond between between the phosphate backbone, or is it a bond between the sugar group in the nucleotide and the nitrogenous base? So what is the nitrogenous base that we are talking about over here? It is either adenine or guanine or cytosine or thymine. Okay, so this is another thing that we are going to discuss about. The answer to this particular question is N-glycosidic bond, right? I'm going to discuss all these other bonds as well, the other kinds of bonding that happens in between the DNA molecules. But if we look at this particular representation, this is one chain of like one DNA strand, and this is an anti-parallel DNA strand. This DNA strand in the representation has a thymine group, which is one of the nitrogenous base, and a guanine group, which is another nitrogenous base. And corresponding to thymine is an adenine in the anti-parallel chain, and cytosine corresponding to guanine in the anti-parallel chain, right? So between these two chains, or between these two anti-parallel DNA, there is hydrophobic bond that is present over here, which is represented by this dotted line, right? And what is the question that we are being asked over here? What is the bond that exists between the nitrogenous group, that is this thymine, and the sugar group, that is this ribose sugar, right? The deoxyribose sugar, for if we are talking about DNA. So what is the bond that exists between this thymine and this deoxyribose sugar, or this guanine and this deoxyribose sugar? So this bond is a glycosidic bond, right? Phosphodiester bond is something that makes up this phosphate backbone. That is also what we are going to discuss in another representation that I have kept for this sake. I hope I have kept it. Just let me check. Yeah, I have. So I hope this particular answer is clear. If not, let me know.
Yes, ma'am. Clear.
Yeah. Okay. So this is a very, very common misunderstanding that we have, especially when we talk about linkages in our DNA. We need to see what is the linkage between which two groups is the linkage being asked, right? If it would have been said that what is the bond between the nitrogenous, between two nitrogenous base pairs in a DNA molecule, what would have been the answer? So the
Can you
answer
please
then,
repeat?
Hydrogen bond.
yeah, so the answer then would have been hydrogen bond, right? So let us discuss this again. There are four kinds of bonds that exist in case of a DNA molecule, right? One is a hydrogen bond, this one that we talked about. The other is a glycosidic bond, which is another thing that we talked about over here, right? In the previous slide. The other two that are left are phosphodiester bond, which is the most commonly talked about, one, and a covalent bond, which is not that commonly talked about. So when we talk about this anti-parallel DNA strands, the hydrogen bonds exist between the two nitrogenous groups. The covalent bonds exist between the deoxyribose sugar and this particular phosphate ion at the 3' site, right? And the ester bond that we are talking about is this bond between the 5' carbon moiety of this ribose sugar and the phosphate ion, right? So since there is this particular ester bond and there is another ester bond above it, these two ester bonds basically form a diester bond, and there is a phosphate ion that is involved in between these two ester bonds, right? So it is known as a phosphodiester bond. So that is it for this particular topic. If required, I will talk about this in detail again, especially the phosphodiester bond. For now, let us come on to question number 10, which is in an RNA molecule, uracil replaces with which one of the following base found in DNA? Right? So which one of these is replaced by uracil in case of an RNA molecule? Is it adenine or cytosine or thymine or guanine?
Thymine.
It is thymine.
Thymine.
And is thymine a purine or a pyrimidine?
Pyrimidine.
Yeah, so it is a pyrimidine. So pyrimidine has a 6 carbon ring, right? Whereas purine has a 6 plus 5 carbon ring. In case of an RNA molecule, the thymine that is present in the DNA is replaced by a uracil. They both have a very, very similar structure. If you can see over here, they both have these two uh, uh, O groups that are present on the same exact carbon, but there is one methyl group that is present in thymine, which is absent in case of uracil, right? So that is the only difference between the structure of thymine and uracil. They are very, very similar in structures, which is why uracil is also so successful in place of thymine in case of the RNA molecule. And this methyl group in thymine is one of the reasons why we say that DNA is or DNA molecule is more stable than an RNA molecule, right? One case is the presence of deoxyribose instead of ribose sugar, because that reduces one reactive group, so it is more stable. DNA is more stable. One of the other features is the presence of thymine instead of uracil, because this is a more stable structure than this uracil, right? So that is another thing we can keep in mind. That brings us to the end of the first part over here. Uh, so we have discussed the previous year's assignment. Let me just shift my focus to the other presentation. I hope it is visible. Anyway, I will once again share my screen. Yeah. Yeah. Okay. So we have discussed the previous year's assignment, with respect to week one problems, right? The week one lectures. Now, the other thing that I told you I will be dealing with is some of the extra questions and concepts. So I have not kept any extra questions actually in this particular lecture. I have had a very unplanned week this time, so I will probably not even take an entire 2 hour class today, which is not the case in the following week. So I will take a 2 hour lecture every other week, but because of some like okay, some unplanned events that went throughout the week, I have not been able to prepare this that well. So instead of these extra questions, I have just kept some concepts that I would like to discuss with you people. Also, I have mentioned over here that it is also for doubt solving for questions in the discussion forum. Like if for the people who joined later in this lecture, I had said that half of my lecture I will dedicate to the previous year's assignment pertaining to questions for that particular week's lectures, and the other half, or more than half, I will deal with some extra questions, concepts, and also I regularly check the discussion forum, if not regularly, at least at the end of the week before coming to this class, before joining or taking this class. I do check the discussion forum, and whatever queries I feel people are having in and they have posted about it in the discussion forum, I try to deal with that concept over here in this particular segment, right? So, I'm so sorry. Huh. So let us begin with this particular extra concepts class. One of the things I wanted to discuss again is the difference between prokaryotes and eukaryotes. Although we pondered upon it a little while we discussed the previous year's question, but I would like a moment to discuss about it in detail again, because there was one of the learners who had some problems regarding this difference, especially between the presence of some membranes between the eukaryotes and the prokaryotes. So that is why I thought I will discuss it once again in this discussion. Just give me a moment. Okay. Yeah. So coming to the first concept, that is prokaryotes and eukaryotes. Uh, we have discussed the features that differ between prokaryotes and eukaryotes. But let us also briefly discuss the origin of these prokaryotes and eukaryotes. So which one of these do you think is the more primitive one? Is it the prokaryotes or the eukaryotes? Which one do you think arose first while evolution was occurring?
Prokaryotes.
Yeah. So the primitive ones are prokaryotes, and the more recent ones are the eukaryotes, right? That is why prokaryotes usually have just unicellular organisms as a part of these primitive organisms, while eukaryotes have unicellular as well as multicellular organisms, which are part of this particular bigger group, right? If we talk in terms of evolution, we can see uh, as you might have seen many other evolutionary trees or evolution depictions of evolution. Here's one of them, which says that there was one a particular common ancestor or a universal ancestor that existed a long time ago, right? From this particular common ancestor, both prokaryotes as well as eukaryotes arose, right? In these prokaryotes, the first probable organisms to arise were bacteria. right? Bacteria are thus also very, very primitive organisms, and they are still flourishing because obviously they do have some advantages and some conditions that they like some additional machineries due to which they can tolerate or withstand really harsh conditions, due to which bacteria are flourishing to this day. There were another class of prokaryotes, that is archaea, which diverged from this and from this universal ancestor, or from this particular ancestry a little later than the origin of bacteria, right? So these archaea are also prokaryotes because they have similar features, that is the lack of cellular like very organized cellular organelles like mitochondria, lysosomes, etc. They do not have a nucleus. They have their DNA in the cytoplasm, etc. Right? And then much later, there was this class of eukaryotes, or the eukarya, that arose. And eukaryotes encompass multiple classes of organisms, such as the protists, which are unicellular eukaryotic organisms. I'm sorry, the protists. Then there are fungi, there are plantae, animalia, all the other organisms, multicellular organisms that exist, are all parts of eukaryotes, right? Moving ahead, I have also listed the cell components, since this was one of the topics that we discussed or that was discussed in the first week's lecture, and there was some uh, confusion regarding what exactly the cell components of prokaryotes are. I have listed them down over here, that the prokaryotes include a nucleoid, which is a the presence of the genetic material in the cytoplasm as a dense material, which is not completely distinct from that of the cytoplasm, because it is not bound by any particular nuclear membrane or nuclear layer, right? Then there is a cytoplasm which has grain-like ribosomes that are present over here. Then there are sometimes plasmids that are present. Plasmids are uh, again some more DNA, like circular DNAs that are present in case of some prokaryotes. These plasmid DNAs are different from the genomic DNAs that are present in these particular organisms. Apart from this, they have plasma membranes, cell walls, and like I discussed in our previous year questions, they often have features from their own mobility, such as flagellum, cilia, pili, etc. Right? In case of eukaryotes, on the other hand, uh, how do these what are the cell components in eukaryotes are? A well-defined nucleus with the genetic material inside of it, then a cytoplasm, which contains many other cellular organelles, such as ribosomes, which are present both as a grainy granulated form, as in case of the prokaryotes, and also on the walls of other organelles, such as endoplasmic reticulum, which like together forms the rough endoplasmic reticulum. Then there are other organelles such as Golgi complex, mitochondria, lysosomes, plastids in case of plants, which are of special types in different plant forms, right? Then there is plasma membrane that is always present in eukaryotes, and cell wall that is present in only the plant cells or some other lower eukaryotes. So these are the cell components of the prokaryotes and the eukaryotes. Then another uh, topic that was discussed upon in yeah, Sanika, did I read your name right? Yeah, you can go ahead. You can turn on your mic and speak.
Ma'am, in
Yeah.
eukaryotes also we have flagella, villi, and cilia, no?
Yeah. Okay, that is also maybe I should have included that in with these plastids. Like there is this class of protists that do have flagella. Then there are cilia that are also present in some of the lower eukaryotes as well. So yes, they are present. Yeah. Okay. Thanks for pointing that out. If there's another question or comment, you are allowed to speak up, right? Okay. So another discussion that was briefly touched upon was model organisms, right? So what are model organisms? Model organisms are basically the organisms on which we do basic biological research, right? And not just basic biological research, even some translational research, like on higher model organisms like mammalian mammals, right? We can even do translational research on these model organisms. Now, why do we need model organisms? Is there a reason? Yeah. So whatever, oh, sorry, I wanted to, uh, why do we need model organisms? So one of the reasons that we need a model organism is that we need to understand a biological process before we can like correct it. For example, if there is a pathological condition,
Excuse me, ma'am, you are unable to be heard. Hello? Ma-
yeah, um, I think I lost the connection. I hope it's back. I'm not sharing the screen currently. I'll do that shortly. Was there a question that I missed?
Ma'am, I was just saying that we were unable to hear you.
I had actually lost connection. Okay, so I'll share my screen again.
Ma'am, one question also.
Yeah. Go ahead.
Ma'am, you just told about translational research. So can you please brief about it?
Okay. So, okay, there is a big debate on what we can call translational research and what we cannot. In my perspective alone, I would not like to cite any examples over here. In my perspective alone, I would list research as translational research when one is working on certain maybe biomolecules or components or compounds or maybe certain biomaterials that is directly going to be used on as it is, maybe just with dosage modification or some such minor changes with respect to dose or with respect to its like bioavailability, etc. It is going to be used as it is, as it has been researched upon in case of a human as well. to maybe sort out some disease, and not just in case of humans. Okay, so why I am a little biased in my opinion about this is because in my department actually there is no professor who works on plants as such, but that is why I tend to forget this. But there are also translational researches, such as, for example, if somebody is deducing a particular compound that can act as a pesticide in a particular plant, and for example rice plant, right, and it can increase its yield, or it can protect it from pests, such that the yield of that particular rice variety is better, and that will help us in long term, because our consumption of rice is that high. In general, people need rice. Or in case of diseases, when we talk about diseases in cattle or in human, or in like our surrounding, if something is wrong, and if somehow there is a deduction which can directly, like that compound that has been created or researched upon can directly benefit an individual that is suffering from a disease, or is maybe undergoing some treatment, right? Then that is generally known as translational research. While what is basic research? Basic research is the like step on which translational research is built on, right? So it is not less important, it is actually more important in some cases, because in order to deduce a medicine, or in order to make a medicine or to treat anything, you need to know what you are treating, right? For example, if you talk about Alzheimer's, you need to know that what is wrong in Alzheimer's that you need to correct. If you know what is wrong, only then will you be able to come up with solutions, that okay, I know that this particular uh, loop or this particular signaling might not be working. So maybe I can give an agonist that can help the signaling to work, right? So for that, you need to know the root cause of any situation. For example, most of the concepts that we study in science are all basic researches, right? So it is equally important, more important in some perspectives, but that is how it is distinguished. So it is more about opinion now. There is a lot of debate that is going upon as to what is translational research, what is basic research, how is one better than the other. Nothing is better than the other. Both go hand in hand, right? Even if we just know about a disease, it is not going to help us if it is not like if that knowledge is like not being utilized to better the condition in that particular disease, right? So both go hand in hand. There is no one good in this case. And it was me who brought upon this discussion upon myself. But I hope it is a little clear, right?
Yes, ma'am.
Uh, Yeah. So again, model organisms is what we were talking about. And my question to you was, before my connection broke down, why do we at all need model organisms? Why? Because whatever answers that we want, we cannot directly work upon uh, individuals, right? For example, if we want a particular, uh, okay, let me just get back to the disease. It is just simpler to explain in that context. For example, if we want to check the like how a particular disease progresses or what causes the disease, right? How are we going to do it? Now, say that this particular disease is affecting individual humans, right? And these humans range from ages of 10 to maybe 80, right? Now, do you think that we will be able to, like earlier, when medicine was just beginning, this had been done by Pasteur and by other scientists at that time because it was a need of the hour that they had reduced maybe a vaccine and they had directly given it to the individuals, right? And some of them worked and some of them didn't work. In cases that it didn't work, these individuals lost their lives, right? Now, at this in this era, is that a chance that anybody is going to take? For example, I have a particular disease and I want to be treated for that disease. And this particular disease that I have does not have a known cause, right? So, can it be treated? The answer will be no. If it does not have a root cause, it will probably not have a treatment, right? In a very rare case, will this scenario work, even if the cause is not known, right? So, for us to understand a cause for a disease, we need some model organisms. Now, what are these model organisms? These model organisms will be mostly organisms that we can handle in the lab, right? With proper ethical clearances. We can handle them in the lab itself. We can breed and we can create a population of these model organisms. And then maybe in our model organism, we knock out a particular gene, right? Which is maybe lacking in a condition that I have. So maybe I have a rare disease in which I lack a gene. Right. This is something that I have gotten through genetic screening. But now, from lacking that particular gene, what is it that is going on inside my body? Who will understand that? Now, for that, we need to assess multiple systems, multiple pathways. For that, we require model organisms. For example, okay, first let me introduce model organisms, then I will talk about some of the utilizations of these model organisms, right? Yeah. Okay. So some of the model organisms that I have listed over here are the invertebrates and sorry, other invertebrates and vertebrates that have been listed over here. Invertebrates such as C. elegans, which are worms, tiny worms, right? Which are mostly clear under a microscope, right? Then Drosophila, which are again very tiny flies. Zebrafish. Then again, there are a class of vertebrates which are popular as model organisms. These include zebrafish, xenopus, which is a kind of a frog or amphibian. Then chicken, which is under the class of aves, mouse, and humans, which are under the class of mammals, right? Uh, yeah. So, and this representation I had copied from somewhere, and that did not include another very important model organism, which is Arabidopsis thaliana, which is a kind of a plant, right? So, why do we need these model organisms? Was our first question. So, in case a human has a developmental defect, right? What is a developmental defect? That when a sperm fuses with an egg and it implants into the womb of the mother, after that, the development of that particular fetus begins, right? During this development, there could be some mishaps that might occur. It might be due to genetic root causes, or it might be due to some other chemical intake by the mother while carrying that fetus, or some totally different kind of a reaction that might have gone on just because of the lack of certain enzyme in the womb of the mother, right? So there could be multiple factors that might have caused a particular defect, and it could be at any time point, like the gestation period for a human human fetus, is around 9 months. So during this 9 months, there is a scope of a lot of developmental uh, changes or defects that could occur, right? But mostly these defects, if they are bound to occur, especially due to hereditary or genetic causes, they occur very early on. It might be carried itself in the sperm or the egg also, right? So there are certain organisms that we need to look at to understand these defects, right? So these developmental defects are mostly looked at in case of chicks, right? Chickens are the most common models. There are other models such as xenobus, wherein developmental biology is studied, but chickens, and why chicken? Because in case of chickens, how do these chickens arise? From an egg, right? So what people do is, the researchers earlier, they found it simpler to just cut open an egg from the top. They make a small window in the egg. The yolk is intact inside the egg, and they just observed the yolk and how it is growing. They are not doing, earlier what they did was they were not doing anything else. They had just made a small window in the egg and they were just observing how the development was occurring. Now that is something that is impossible to do in case of a human or a mouse, or some other organism, right? But over here, since the fertilization was external, sorry, fertilization was not external, but since the development was external, it was easier to observe, right? Similar in case of xenobus, which has eggs that are laid outside, right? So they are not carrying children in the womb. Therefore, the development is external, outside of the body. So it is easier to monitor that development. And now, with advancement in science, people have been able to make certain modifications in these developing eggs also. For example, now since we understand that, okay, this region of the egg, once we form a window, we do understand from existing literature that, okay, this one particular region of the yolk, that after 4 days has become this big and it is adjacent to this particular region, is going to form the head of the chicken. And we want to form a developmental defect in this particular portion. So we maybe introduce some bead containing retinoic acid over here, right? Or some other compound that might cause a defect. And we see that what is the kind of developmental defect it causes. And if once this chicken has basically hatched, or once it has grown, uh, first is, will it grow if such kind of a defect is there? Second, if it has grown, what is the kind of morphology that we see now? Is it similar to any existing human conditions that we know of, right? So this is how model organisms work. For example, if we devise a new cancer therapy, do you think it will directly be tested on a human? No. First, it has to be tested on other existing model organisms. For example, some kind of mice, right? Some strains of mice, which might have an induced tumor inside them. So how do these cancer therapies actually work is that people, they usually choose this particular model organism. There are Drosophila also that are used in case of cancer studies, but they are basically for the not for translational research, like I mentioned below. For translational research purposes, mostly mice are used, and even for basic research they are used. But what happens in case of cancer therapies is that these mice are taken, some particular either immunocompromised mice, or like immunocompromised mice, because what we do with these mice is that these inbred mice, under ethical clearance, what happens is that you can basically take a portion of the tumor from the human through biopsy, and you can culture it. And then after it has reached a certain time point and a certain mass of cells, you can introduce these cells into the immunocompromised mice. Now, why are these mice immunocompromised? So that their body will not react that harshly to anything that is foreign and it is being introduced into their body. And what will it result in? It will result in a tumor in whichever location this particular tumor has been injected in case of the mice, right? So now this particular mice with this human biopsy sample that had been cultured has a humanized tumor that is induced into it. Now we can use maybe pre-existing drugs or repurposed drugs, maybe that has already been used in some other type of cancer, some other kind of disease. Maybe we can just check whether that compound, if we give it in this particular region on the tumor, will it help in shrinkage of the tumor? Will it help the tumor to disappear? Or will this drug shrink the tumor, but it will lead to relapse of this cancer again in this mouse, right? So that is how model organisms are being utilized right now. There are other studies such as genetic level studies that you might have already read about in your genetic lectures that are usually done with the Drosophila, right? Or with these plants such as Arabidopsis thaliana or pea plant in case of Mendel, right? All these are model organisms. So I just wanted to give you a brief introduction on model organisms. And I actually wanted to include some points over here in writing as well. But as I told you, I am a little underprepared for this lecture, but I can, if you want me to, in the upcoming lecture. And if you have any doubts regarding this particular slide or regarding whatever I said, please let me know. I will try to clear it up. Okay. So, if not, we will move on to the other like separate topic that I wanted to discuss, which was amino acids. And why I wanted to discuss this would become clear in maybe the third slide of amino acids when I come on to that slide, right? So, what are amino acids basically? Just let me take a short. Okay, I hope that. Oh, I'll not take any chances. I'll just stop sharing and share again. Yeah. Okay. So the first thing that I wanted to discuss in this particular respect was the basic, which is what is amino acids, right? So, and why is it named so? It is named so because these biomolecules or these structures have an amino group that is present, that is the NH2 group, and a carboxyl group or COOH group that is always present in these biomolecules. Along with that, there is a CH that is present, and along with this CH, there is one carbon covalent bond that still remains, right? So carbon can form 4 bonds, 3 of which are occupied now. We know that one has a hydrogen, one side of one, sorry, hydrogen is bound to one of to the carbon. The other side it is bound to a carboxylic group, and on the third side it is bound to an amino group. The fourth side is the side chain, right? This side chain can vary, and the variation of this side chain determines which particular amino acid this is, right? So there are 20 to 21 common amino acids that people generally talk about. This one is a representation. This image I had taken from Lodish. I think this talks about 20 common amino acids. Some representations include another special amino acid over here, which is selenocysteine, which is a little different in structure from the cysteine, right? And how these people have divided amino acids over here. in the 8th edition of Law Dish is that there are some hydrophobic amino acids. There are some other hydrophilic amino acids. These hydrophilic amino acids can either be basic or acidic or polar amino acids with uncharged side group, right? Or there could be some special amino acids. Now, there why are these called special amino acids is something that I think you can ponder upon, you can read upon it. It will help you. But to give you a brief overview about it, all these amino acids are building blocks of proteins, right? We know that each amino acid is going to be bound into a chain of amino acids, which is ultimately going to form a protein, right? So that is the basic function of these amino acids. And there are some special amino acids that are mentioned over here. Why are these amino acids particularly special? Because they have some additional properties. For example, cysteine has this thiol group, the SH that is present over here is a thiol group. And what this does is that basically it helps in formation of disulfide bonds between protein structures. And these disulfide bonds or chains are very, very relevant and very, very important, especially when we talk about some structural proteins, right? So, if you might study a little more upon proteins, you will know that a lot of properties of the protein are brought about by the structure of the protein, even more so than the sequence of the protein, right? Even if the sequence is different, but the structure is same as the original protein, it will probably work in more cases than not. Right. So the structure of the proteins are very, very integral. And disulfide bonds are linkages that really help and are really relevant in maintaining many such structures. Right. Then glycine is listed as another special amino acid. This is because it is the smallest amino acid. As you can see, the side chain in this particular amino acid is just another H. So it is the smallest amino acid that is present. And because of its small size, it is very, very versatile. So if glycine is present, probably around that particular base, that is glycine, the entire protein can twist and turn and rotate around this particular amino acid, right? So it provides flexibility to the protein structure. On the contrary, proline, if you can see, there is some ring type of a structure that is present over here, right? Because of this ring type of a structure, it provides a very, very rigid kink wherever it is present in the protein, right? So, and around this, the protein cannot really rotate. So it will provide a very a rigidity to a particular structure in the protein. So that is why these are some of the special amino acids. And it is a very, very huge discussion, just amino acids. But the names is something that you can go through. Along with names, there are 3-letter abbreviations of each amino acid. For example, for leucine, it is LEU. For methionine, it is MET. For aspartate, it is ASP. And there are even single letter denotions that have been given to these different amino acids. For example, A for alanine, R for arginine, H for histidine, then E for glutamate, etc. Right? So you can go through these once. Yeah. Apart from that, there is this another representation of amino acids that I have put. Now, what is different in this representation from that previous one? In that previous representation, the structures of amino acids were listed, right? So that is for when you need to go and look at the structure of these amino acids, some of which are important. I personally have not been able to recall most of the structures after having studied biochemistry for so long. But that is my shortcoming. I am not very good at chemistry per se. Uh, but this is something that you will need to have a hold on. Uh, yeah. So when we talk about amino acids, not just their structure is relevant, but there are other features that these amino acids do. For example, these amino acids are present as a triple codon, right? As a triple code. These triple codes will basically tell us uh, uh, which particular amino acid is to be recruited. Now, what are these codes? These codes are the nitrogen bases or the groups that are present in the DNA molecules, which are transcribed into the RNA. And now these RNA, when it is translating into a protein, it will use this particular sequence to recruit which particular amino acid has to come and sit at this place, and which amino acid, a particular amino acid has to follow this first amino acid, right? So this is the code for that. Now from this table, you will see that some of the amino acids are highlighted. For example, this AUG is given in red and it codes for MET, that is methionine, right? So AUG, why it is highlighted is because every time the protein translation begins, it will begin with this sequence, that is AUG. So the first amino acid that will always be present in every protein is going to be methionine. This is where it starts. The other three that are highlighted are the stop codons, that is UAA, UAG, and UGA. Now, these stop codons do not code for any amino acids, right? So, whenever the protein forming machinery, that is the ribosomal subunits that are present and the tRNAs that are present with it, whenever they will encounter this particular any of these particular codons, what they will do is that they will not recruit any other amino acid, and the protein translation will stop at that point, right? Apart from this, there is another important thing that you might notice over here, which is that, for example, if we talk about threonine, there are four codons that are coding for threonine is written over here, right? Serine, four codons are coding for serine. For tyrosine, two codons are coding for tyrosine, right? Then there is where's tryptophan. For tryptophan, only one codon is coding, right? For methionine, also only one codon was coding, right? So this means that there are more number of codons than the number of amino acids. We have how many amino acids? Mainly talking about, we have 20 major amino acids, right? It can go up to 21 to 23 if we include some of the less common amino acids as well, right? So majorly speaking, we have 20 amino acids, and these the number of codons that we have, how many? So we have 4 into 4, 64 codons, right? So for 64 and minus 3, if we, because these do not code for any amino acid, right? 61 codons we have for 20 amino acids. So this means that there will be some codons that will code for multiple codons that will code for one amino acid also, right? So that is the case that we see over here. Now, what is this known as? This is known as degeneracy of the code, degeneracy or redundancy of the code. This means that there are some, again, what we said, there are some codons that are going to code for the same, like there will be four or five variants that are going to code for the same amino acid, right? That is what is degeneracy. Now, over here, when we talk about these, there are some essential amino acids, some essential codons that you need to remember. One was AUG that codes for methionine, and it, as I told you, it always is the first codon to be present in any protein translation or any protein chain. So AUG, sorry, is the first one that you need to remember. Then another one that you need to remember is UAA. UAG and UGA, right? All of these code for stop codons, right? All of these code for stopping of the protein translation. And along with this, one other important one, even if you don't remember most of the others, one important one that you also need to remember is tryptophan, that is UGG. Why do you also need to remember this is because AUG for methionine and UGG for tryptophan are the only two codons which do not have any degeneracy in them, right? These are the only two codons that code for these two amino acids. So if these two codons are not present, these two amino acids will not get encoded, right? It will be replaced by some other amino acid, or it will be replaced by a stop codon or something else, right? So moving on, I hope this portion was clear. Now coming on to why in the first place was I discussing amino acids, right? So it was for this particular hypothesis that I wanted to discuss. First, let us see what I have listed over here, that there exists degeneracy in the genetic code, right? Hence, some tRNA molecules can recognize more than one set of codons differing at the third codon only. Can you understand the statement? How we can look upon it is through examples. For example, if we talk about serine over here, you can see that the first two codons over here are same for serine. Only the last codon is varying, right? Similar is the case for proline. I just lost my pointer. Wait. Yeah. Similar is the case for proline over here, right? The first two codons are the same. There is difference only in the last codon. For histidine as well, right? So this degeneracy, it generally occurs at the third codon. For this reason, there is a hypothesis that exists because of some of the observations that had been made by scientists, which was that not as many tRNAs are present as many codons are there, right? Now, why is this so? Because there is a hypothesis which is known as wobble hypothesis that has been put forth by the researchers. This hypothesis states that fewer tRNAs can help in translation of mRNA because of the degeneracy of third codon that is present, right? So it means that once a tRNA that comes over here, tRNA looks a little like this, right? So once a tRNA comes over here, the first two bases is what it actually has to recognize in the mRNA. The third base of the mRNA, even if it is not the same. For example, the anticodon on this tRNA is maybe A, G, A, right? If it is AGA, means it will recognize the codon UCU, right? But even if it is UCC or UCG or UCC, UCG or UCA, it will be able, this same anticodon containing tRNA will be able to recognize this as well, right? This is known as a wobble hypothesis. I hope it is clear. If not, please tell me. I will try to explain it further. Else I'll understand that it is clear. Okay. So again, this was answering one of the doubts that I saw on the discussion forum. That is like one TR. It was something like this only, that are there multiple tRNAs for multiple like codons, or can one tRNA recognize more than one codons? I do not really remember the statement that was given, but I this was the concept that was behind it, right? So another thing that I briefly wanted to ponder upon was metabolism. So what is metabolism? Metabolism is the set of life-sustaining chemical reactions that is present in an organism. It is a set of chemical reactions in the body cells that change food into energy. So, what is our metabolism? Whatever food we are intaking, how it is being converted into the energy that we need. That is our metabolism, right? So, uh, our cellular health, our health at the cellular level will also determine our metabolism, right? So this is a very, very commonly spoken about topic. This is the chemical reaction that goes behind it, that whenever we intake food, right, from that food, uh, it's like breakdown or digestion. It helps in condensation reaction of ATP and a free phosphate into ATP, which is a high energy molecule, right? This high energy molecule is basically prepared whenever our food is being consumed and utilized and formed into energy, right? Once this high energy molecule is prepared, whenever in future we need energy, what will happen is there will be a hydrolysis reaction that will occur. What will this hydrolysis reaction do? It will basically break down this phosphodiester bond, right? By breaking down this one phosphodiester bond, it will remove one phosphate back, which will become free, and ATP will be left. And in this process, the breaking of this bond will release energy that is basically going to drive whatever process uh, this energy was required for. For example, it could either be for sodium potassium pump functioning, where ATP is required because it is an active transport, etc. Right? So that is uh, about metabolism. Now, why we discussed metabolism was to come to the energy currency of the cell. What is the energy currency of the cell? We will discuss about that. But first, let us talk about uh, how a particular nucleotide is basically formed. So a nucleotide, which is ATP is also one of the nucleotides. That is why we are discussing this, right? So let us see how this nucleotide is formed. So first, we just have a base, ribose, which has a ribose sugar, and it has a nitrogenous base, which could either be any of the purines or the pyrimidines, right? Which are both linked by a glycosidic bond. And then along with this sugar, sorry, just these two, the base and the ribose or the deoxyribose sugar will be known as a nucleoside, right? Then if this nucleoside has one phosphate attached to it, it will be known as a nucleoside monophosphate. If it has two nucleotides attached to two phosphates attached to it, it will be known as a nucleoside diphosphate. And if it has three phosphates attached to it, it will be known as nucleoside triphosphate or a nucleotide, right? Now, why we were talking about this was because uh, one of these nucleotides, that is nucleoside triphosphates, with one of these bases is the energy currency of the cell. Now, which one of these is that? These are the four nucleotides. I have intentionally not mentioned the names of these nucleotides, but I have given a hint over here because the structure is provided. Which one of these do you think is the energy currency of the cell?
Ma'am, the first one.
Yeah. So it is the first one because over here, the base that is present with this with this deoxyribose and these three phosphates is adenine, right? So this particular one is ATP. Right now, this one is GTP, and this one is TTP. I think no, TTP has a methyl group. This one does not have a methyl group. So the one that does not, this one is UTP, and this one is CTP. Right. So ATP is actually the energy currency of the cell. Now, one of the things that I thought I will discuss today in the lecture, but I have not included in the further slides. So it could be an open question for today, wherein I will end the lecture and I will discuss it next time, or I can briefly discuss it today. Is why ATP and why not any other nucleotide is the energy currency of the cell? Because what did we say? That what is required for the energy? Is it the base that is giving the energy?
No, ma'am. It is the
No,
phosphate group.
Yeah, so it is the breaking of this phosphate group that is giving the energy. So why is it so that only ATP is the energy currency of the cell and not any other nucleotide? So although I am introducing this question because it is one of the questions that I think is also very commonly asked in a lot of interviews, and it is just to test the curiosity of the student. There is no one direct answer to this question, but there are a lot of speculations as to why this could happen, right? So I will also not give you one direct answer. I will tell you all those speculations. Uh, but my question is whether you want to briefly discuss it in today's class, or should I, with pointers written down, discuss it with you in the next lecture? Do
yes,
you want
ma'am.
to
Can
read
you
about it?
please give us the time to research about it?
Ma'am, you can discuss it today only.
I can? Okay. Okay. Anyway, you can go and you can research about it. So since I said that it is also another question that does not have one direct answer, right? So it just has speculations about what could be the reason. Okay. So, okay, some of the points that I can discuss. I think I don't think I have any other slides, so I can discuss some points as to why it could be ATP and why not any other of these TTP, UTP, GTP, CTP, why not any other, right? So one of the reasons that is often cited is that during evolution, when this energy currency was being devised, this might have been most abundantly present, right? ATP might have been most abundantly present, due to which what happened? It could happen that at that time it was abundantly present, and now maybe GTP could have been abundantly present. So, why is, if at that time ATP was abundantly present, why is it still dominating? Could there be a reason for it? Okay. So what could have happened was that initially uh, the the systems were a little simpler, right? Although bacteria, protists, etc., also have very complex machinery, but as compared to eukaryotes, the systems were a little simpler earlier, right? So at that time, when this energy currency was being evolved or devised in the first place, if ATP was abundant, all the other mechanisms or all the other machineries, for example, other enzymes, etc., would have like been formed, that were being formed at that time for the first time. For example, certain kinases, certain proteases, etc., right? They would have had a specificity for ATP, right? Now, uh, even if enzymes, for example, are very, very specific for their substrate. So as I said, that structure in many cases is more important than the sequence, right? So if an enzyme has a particular site, an allosteric site, right, wherein ATP can only bind, do you think GTP will be able to go and bind? No, right? So if at the time of like evolution, ATP was present in abundance, the enzymes that might have also evolved along with it might all have this specificity for ATP, right? For the structure of ATP, which is why, and since these are very, very specific for their substrate, like I said, they will not go for any other currency in exchange for ATP, right? They will not be sufficed with the GTP or CTP or TTP, etc. Right? So that could have been one of the reasons. Now, what could have been another reason? Any speculations? Okay.
Ma'am,
So
it
another,
can be that
huh,
it
hmm,
can be that the double ring structures are generally more stable than the single ring structures. And in that case, if we compare adenine and cytosine, the cytosine has the carbonyl group that is much more reactive than adenine, which makes adenine more stable than cytosine as well.
yeah. So that is another very, very good point, right? So because this was a purine, right? And purines were thermodynamically supposed to be more stable than pyrimidines, like other of these other groups, right? But again, uh, what are the two purines that we know of? It is adenine and guanine, right? Yeah. So adenine and guanine are the two purine groups that we know of. Just to revise, okay? There are two purines that we know of. One is adenine, another is guanine. So thermodynamically, yes, these both might have been more stable. Out of these, again, guanine, adenine was chosen over guanine, right? Now, is it so that guanine GTP is not used anywhere? Like I don't know how many of you might be new to this particular subject, cell biology, molecular biology, etc. But if at any level during your undergraduation, not during your school time, but at least during your undergraduation, or if anybody of you are master students during your masters, if you have red cell signaling, you would be aware of uh, GPCRs, that is G protein coupled receptors, which is the largest class of membrane proteins. And it is also one of the highly targeted molecules for drugs that are administered in humans for most of the diseases, right? So they are GPCRs. Now, these are G protein coupled receptors which work with GTPs and not ATPs, right? So, GTP is also one of a very significant um, this uh, nucleotide, and it is used in very, very specific facets of molecular biology or cell biology, specifically for protein synthesis or protein breakdown or signal transduction, etc. Right? So it could also have occurred that to basically divide the roles of ATP and GTP. ATP remained the energy currency of the cell, while GTP took on more specific roles for signal transduction. It is one of the major players in signal transduction, okay? For signal transduction, protein synthesis, etc. Right? So that would have been the third point. So as I told you, that will be the end of today's discussion. If there is any question that is pertaining to today's class, you are most welcome to ask it. Even if it is not pertaining to today's class, if I can answer that question, I will really try to. If not, I will always come back prepared in the next class with an answer for your queries. So you can ask any queries if you do want to. Um, if not, that will be the end of today's lecture, right? So yeah,
Excuse
that is
me,
the
ma'am.
basic. Huh. Yes.
Ma'am, actually, I just want to ask that I was sent the NPTEL mail in which four links were provided for four different sessions. So
Yeah, yeah, yeah.
all those sessions are different or they are same only?
So, actually, you will have to visit at least once every session to understand how it will happen. But yes, they are similar with respect to this, that there are four different TAs that actually are taking similar kind of sessions at different time points. Now, the uh, things that they might be teaching are different because we amongst ourselves are not coordinating, because we are at different time slots. So it is more about which learners are available at which time slot, right, to attend these lectures. So we all have the same previous year's assignments, so that will be common in all the four TA lectures as per my knowledge. But apart from that, how the course is being dealt by these TAs is up to them completely. So for that, you will have to, like, at least maybe once a YouTube link is uploaded, you can go through that, or once you can like log into every class and look through that. So for that, you will have to see that.
Ma'am, you will be available at this slot every time.
Yeah, yeah, yeah. So I will always take the lecture for this particular subject at 3 to 5 every Sunday. So that is actually going to remain constant. So like 4 to 6 lecture on Sunday is going to be taken by the same TA every time. We will not interchange our slots because like we have also uh, work schedules. According to that, we have scheduled this on at a time wherein we can actually uh, prepare and we can give this lecture. So we are not going to change these time slots. And if at all there is some emergency due to which any of the TA has to change a time slot, you will get a mail from NPTEL regarding it. Yeah.
And ma'am, one more thing I wanted to ask, if you feel
Yeah.
comfortable to answer.
Yeah. Yeah.
Actually, I just wanted to know about small journey of yours to PhD here at IIT Kanpur. I am from Delhi University, first year microbiology student. So it will be a good direction for me
Masters.
to work further. No, no, ma'am,
Okay.
BSc, bachelor's.
BSc. Okay. My bachelor's is also from Delhi University. I have done my bachelor's in zoology actually. And I have actually had a background of, um, I have had a zoology background before joining my PhD. So my bachelor's and master's both have been in zoology. And yeah, so what, like, if you are in your first year and you are this, like, you are that much aware that you are even attending these courses, because frankly, to speak about all these, by the end of my graduation is when I was getting aware of like the things that I actually should do if I wanted to get into research and all. But being in your first year, I think it is a very good point to start, and for you to interact with the other scholars also. So one thing that I would really like to suggest to you is that I don't know whether you are in a college or in the department, like the central department. I don't know how exactly you have been allotted. Are you under a
College.
college? Okay.
Yes,
So
ma'am.
does
Bhaskaracharya
your
College.
college also have a research lab? Like, are there professors who actively work, uh, or do they just like, are they more uh, the teaching kind of professors?
Ma'am, more towards teaching.
Okay. So in that case, whenever you get a summer break, no, during that time, what you can do is, if you live in Delhi, then I would suggest you to speak to your college, like professors in your college who might have okay. Uh, Most of my professors were also students from Delhi University while I was in an undergraduate there. So they all had some labs that they were affiliated to, right? And I was in the North Campus. So they all had some or the other lab that they were affiliated to. So some of them were working in my department as well, like not a full-fledged uh, uh, maybe topic of research, but they did try to do like shorter research studies. And if not in the department themselves, they were affiliated to their their labs from which they had graduated, and they had always encouraged us to like work in those labs and to gain some experience, do like learn some basic techniques like cell culture, if that is allowed or not, then PCR, some basic hands-on techniques. It will really help with your CV, and it will help you to understand to some level the scenario of research in India, right? Although that scenario at a university level is much different than that at a research institute, but like it will help you to gain some points for your CV. Then it will help you gain some uh, like not everybody is interested in research in the long term. I have had a lot of colleagues who weren't interested, and I also have had a lot of colleagues who never had the chance to assess whether they were interested or not until they joined PhD, right? They had never had hands-on or wet lab training with a research point of view. So I would really encourage you that at least my professors back in DU were very helpful in this regard, that at least in summer, in your summer break, maybe you can join some short internship in your college as well, in your college or in the main departments, any of the department, be it microbiology or genetics or biochemistry, any of the departments. Maybe you can talk to some of your seniors for that as well, who might already be in those labs. You can approach people via LinkedIn. For now, this is what you should do, being in your first year of undergrad. And it is a long way to go from there, right? You have to choose your master's discipline. You have to choose whether you want to continue in India or not. You want to go abroad if you want to do research or not. So all that comes much later. But get some hands-on experience with these techniques if possible. That is what I can say.
Thank you, ma'am. Actually, two major things that I also want to say is that first of all, I want to continue in India only. I never want to go abroad. And second is,
Okay.
though I am interested in lab work, and we usually have many projects going on in our department in the college, but I am more interested towards professorship in future.
But okay, that is actually a very valid point. But to be a professor in science, so this is also something you will ultimately understand once you are maybe in your third year or your master's. To be at good universities in India, if you want to remain in India, to be in good universities and to be professors in those universities, there will be a very, very basic requirement of a PhD, right? So there are very less places wherein you will get a good position as a professor or as a permanent job as a professor without having done a PhD to be the minimal qualification, because there are candidates who will have done postdoc from institutes within and outside of India who will be fighting for the same positions, right? So there will be some heat that you will eventually have to pay for that, pay to that.
Yes, thank you very much for the guidance, ma'am. Means a lot.
Yeah, sure.
Hello, ma'am. Ma'am,
Yeah,
I'm from Calcutta University and
yeah.
first year of my undergraduate biochemistry honors. And ma'am, I'd like to know that how should I like start reading research papers and where should I read them and what research papers should I read? Like,
Okay, so do you have any such course like research methodology or any such? Do you have any such course?
no, ma'am.
Okay. So actually, getting in the habit of reading a research paper is a very, very kind of tedious process because you will only understand the value of those research papers once you are actually working in that field, right? You will have that curiosity to know about, okay, I was also maybe thinking about going in this direction, and that this person has published and this might validate what I was thinking. And that kind of research, curiosity, you will have once you are doing something like that. But a basic way to start reading such research papers is uh, there are some major journals that are more popular than others, maybe. For example, Nature, Cell are some of those, right? So Nature has one, most of these journals have their own news and views section. Nature is one of the journal which has a very like lively news and views column, right? So you can just search Nature news and views whenever you are surfing on the internet next. So what that gives you is it opens a page wherein there are multiple headlines like you get when you open in shorts or any news apps, right? There are multiple headlines that you get on that. You can just click on those links, whatever you are finding interesting. So it is not that Nature news and views will only have news from papers that are published in Nature. It will also have news from good research papers published in Science, in Cell, in JCB, in other PNAS, and other journals as well, right? So they will basically have some cutting edge research articles. And what this news and view will do is that they will have written a short article. Like there is a research paper and there is an article on that research paper, right? So they will have written these articles on that research paper so that it will be a little easier for you to understand that particular research paper, what it was aiming to do, what is the outcome, like how basically uh, what is the significance of the research that has been done by this article, was what was the question asked, what was the answer that was obtained, what are the future prospects of this research. All these things will be written in the form of an article, which is a little simpler than the actual research paper, because for the actual research paper you will have to understand uh, techniques, right? For you to interpret Western blots, you will have to like sit and see, okay, this is the pattern. Like this is the principle of Western blotting. This is what happens after a Western blot is obtained. This is a representation of that Western blot, and this is what they are trying to tell using this. Or if there is a staining image, then this has been stained by this. So it is a little complicated to understand a research paper than an article on that research paper. So first you can go and read that article, right? If you are interested, at the end, they will have given the link for that research paper. You can open it up and you can read it, right? But even just by reading those articles, you will stay up to date upon some of the research that is going on around the world. And you will also like understand what is the kind of research that you are more interested in. That okay, I might like reading more of these developmental biology papers. I might be more interested in reading some of the computational papers. So that will also help you to narrow down what kind of research you are more interested in, right? So that I think is where you can start.
Okay, ma'am. Thank you. And ma'am, another question I want to
Yeah.
ask is that how do I get summer or winter internships in
For
Calcutta or
that, you will
maybe
actually
outside
have
Calcutta?
to, you can talk to your professors and your department or your institute, or once you know, or even if you are trying to figure out what kind of a research you are interested in, you can actually go and check out profiles. Like, for example, if you live at a particular city, for example, Kolkata, to just as an example, if you live in Kolkata, there are multiple institutes that are there in Kolkata, right? For summers, you might want to visit home. You might not want to stay back at your university, right? So you can go home. Simultaneously, you can maybe mail professors from IICB, from Bose Institute, from other institutes in Kolkata, right? And you can go on the websites of these particular institutes. You can look at what professors are there in the department. You can look at what kind of research they are doing. The research areas are always mentioned. Sometimes some detail about the research is also mentioned. You can look at that and you can directly write to that particular person, right? If you do not have a direct contact, like a senior, uh, that or like I don't think seniors will be able to help that much, because you will eventually have to write to that professor. So a professor in your institute can help you get a fellowship if you approach them, and if they already have some work going on in some of the lab, or some other lab, or you can directly write to the person that you are interested, in whose lab you are interested in working in, right? So you will have to, like, it is not always compulsory that you will write to one person and that one person will reply to you. So keep 5 6 options, write to 5 6 people, see who replies back, and accordingly decide.
Okay, ma'am. Thank you. Thank you so
Welcome.
much. Ma'am, I
Welcome.
enjoyed your session very much.
Thank you. Thank you. Okay. So that will bring us to the end of the session for today.